Hello and welcome to the last video of the semester, or the trimester. This is the second part of chapter 15, Special Senses. Okay, so the olfactory receptors. This is the first cranial nerve, olfaction, and we can see the olfactory tract and then the olfactory bulb penetrating the ethmoid bone to lay down its receptors in the nasal cavity, in the nasal conchae. And these receptors then sample the air as it comes in. There are a number of bipolar olfactory epithelial cells that you can see that are responsible for transduction of these smell receptors, activation of the olfactory cilia, and increased action potentials down these sensory neurons. Now, the odorant transduction pathway utilizes a GTP binding protein, and in this mechanism the odorant binds to the receptor, and this activates GTP to then bind to adenylyl cyclase, which is going to create cyclic AMP, which is then going to open up certain ion channels and elicit a response. Now, the ions are going to flow. These are going to be sodium and calcium, are going to cause depolarization and activation of this olfactory cell. So if you look at the tongue, you know, they say taste is 90% smell. Well, let's look at taste for a second. So we've got these different papillae, foliate, papillae on the outsides, fungiform on the front, the middle. And the papillae are tiny raised protrusions of the tongue that contain the taste buds. Now, there are 4 taste buds. There's filiform, fungiform, foliate, and circumvallate. And all of them, except for the filiform, allow to differentiate between sweet, salty, bitter, sour, and umami. In the posterior portion of the tongue, you have your tonsils, lingual tonsils, as well as your palatine tonsils, which are the ones that are usually removed. So the papillae, or the taste buds, here's an enlarged section of a single taste bud. You can see the nerve with the sensory cells here. So the lingual papillae are small structures on the upper surface of the tongue that give it its rough texture. And then here is an up-close picture of the taste cell, the gustatory epithelial cells, as well as the gustatory hairs. And you can see the first cranial nerve coming in, innervating this epithelial cell. So this would be on the tongue. This would be one of the taste buds. Now, the tongue is actually innervated by 3 different cranial nerves. The facial nerve, which is 7, the glossopharyngeal, which is 9, and then the vagus innervates the posterior portion, probably mostly a gag reflex, and that is 10. So these signals merge into the medulla, into what's called the solitary nucleus, where they then signal up to the thalamus, into the gustatory cortex. Okay, let's take a look at the ear. So the outside of the ear is called the pinna or the auricle. You get your lobule, which is where earrings go. Inside there's an external acoustic meatus. This meatus goes through the skull bone. This is your external ear. Now the middle ear is where the acoustic meatus arrives at the tympanic membrane, or your eardrum. And then there are three small bones responsible for transduction of the sound to the cochlear nerve. And then the inner ear is a labyrinth, a cochlea, which detects acceleration, deceleration, as well as pitch of sound. Now the tube that then goes to the throat is the pharyngotympanic, or the auditory tube, or it's sometimes called the eustachian tube. Okay, so if we look at the tympanic membrane, this is what's going to first transduce the sound and activate these little auditory ossicles. But we've got a malleus, which is the hammer. We've got an incus, which would be the anvil, so the hammer beats on the anvil. And then there is the stapes, or the stirrup. This is then connected with the vestibulocochlear nerves. This is the middle ear with the bones. The inner ear is this labyrinth with the semicircular canals and the cochlea. And here is that pharyngotympanic auditory tube. There is a muscle in the ear called tensor tympani, and its role is to dampen the loud sounds, such as chewing, for example, or shouting, or thunder. But it doesn't have a very fast reaction time, so it can't protect against hearing damage with, like, gunshots and explosions. Those is kind of caught off guard. So the tensor tympani muscle is attached to the malleus, where it dampens the sound. It is pulled away from the incus so that there's no longer transduction of this wave. There's also a stapedius muscle connected to the stapes that has a similar action. When it is contracted, it will dampen the sound that comes in the ear. Okay, and the inner ear is this membranous labyrinth, and there are this is the cochlea, and in the cochlea there's this labyrinth. So it's folded up. If we unfold the cochlea, which I'll show on a subsequent slide, we can see the purpose of this long organ. But you also have the semicircular canals, which pick up X, Y, and Z axes, or movement on the X, Y, and Z axes accordingly. And this would be participating in balance. But the facial nerve, the vestibular nerve, the cochlear nerve, these are all nerves that innervate the vestibulocochlear apparatus. Inside the cochlea is modulus. It's a conical-shaped central part of the cochlea, and there's some bone in here, as well as the cochlear nerve. And if we zoom a little closer, we can see these chambers contain fluid called endolymph. So the cochlear duct, or the scala media, contains endolymph, and this endolymph moves as the body is moved. Excuse me. and it will activate hairs, which will increase action potentials. But then we also have two other containers, the scala vestibuli and scala tympani, which contain perilymph, which is very close to endolymph. Now, the perilymph and the endolymph have unique ionic compositions that allow for electrochemical transduction to occur. Scala, as in scala vestibuli or scala tympani, refers to the ladder, and so there's just this laddered kind of shape that you see with the scala vestibuli and the scala tympani. Okay, another look at the cochlea. We see two different membranes, the basilar membrane and the tectorial membrane. So the tectorial membrane is a membrane of the cochlea, of the inner ear, and it extends along the longitudinal length of the cochlea parallel to the basilar membrane. It's innervated by the cochlear nerve, a number of hair cells with supporting cells, which are going to send afferent nerve impulses back to the brainstem. There's endolymph and perilymph that flows through this cochlea, which then will displace the hair cells, leading to action potentials. Now, there are a few bony labyrinths that I want to make you aware of. First of all is the receptor region of the crista ampullaris. So this is the organ of rotation. Okay, they're found in the ampullae of each semicircular canal of the inner ear, meaning that there are 3 pairs in total. Now, the function of the crista ampullaris is to sense angular acceleration and deceleration. Then there's the macula, not to be confused with the visual macula. This is the auditory macula, which has a utricle and saccule. And then there's the spiral organ, which is the cochlea itself. So the macula is going to be for equilibrium, just like the crista ampullaris will be for equilibrium, rotational and angular acceleration, whereas the spiral organ is going to be for hearing. So let's just talk about sound for a second, but we have the tuning fork, when activated, produces a sound wave with areas of high compression and then areas of rarefaction, where the waves are separated. And so to look at the sound wave, we've got a trough and a crest, an amplitude, a wavelength. And so as sound waves radiate outward, they're activated, or they activate movement of the endolymph and perilymph, and then activate the cilia. For sound, pitch is to increase the frequency. Okay, so a higher pitch noise would be an increased frequency versus a lower pitch noise, which would be more spread out frequency, lower frequency. There's also loudness, which would be straight up amplitude. So you can increase the loudness of something just by increasing the amplitude of the sound wave. So if we were to take the cochlea and unroll the basilar membrane, we see a long ladder-like apparatus that starts with short, stiff fibers, which are going to be activated with higher frequency, and then long, floppy fibers, which will be activated by lower frequencies. This is how the cochlea detects sound. And so you detect the sound from 20 kilohertz down to 20 hertz. Now, bending the stereocilia opens or closes the mechanically gated ion channels in the hair cells. So with the basilar membrane at rest, we have very little activation of this cochlear nerve, maybe a few action potentials, but as the hairs pivot towards the tallest hair, you get mechanically gated opening of these ion channels, more ions flow, more neurotransmitters released, and you have increased action potentials in the cochlear nerve. When you pivot away from the tallest hair, you have the opposite, where these action potentials are shut off. The cochlear nerve then synapses in the medulla, where it radiates up to the thalamus and then into the primary auditory cortex. And this is in the temporal lobe. So we see our bipolar afferent cells, these receptors that are activated. You've got your spinal interneurons. This is in the brainstem. You get the medulla. It then throws projections up to the thalamus, to what's called the medial geniculate nucleus of the thalamus. And then that throws another signal into the primary auditory cortex in the temporal lobe. So the membranous labyrinth of the of the inner ear, you can see the three semicircular ducts and canals, and there's anterior, posterior, and lateral, and these are going to detect Z, Y, and X axes. You've got a number of cranial nerves coming in, the facial nerve, the vestibular nerve, the cochlear nerve. You can see the utricle in the vestibule, as well as the saccule. Now the macula, not to be confused with the visual macula, but there's a macula in the inner ear. Excuse me. So there's a macula of the utricle and a macula of the saccule. Now in these macula are canicilium. The canicilium is a special type of cilium on the apex of hair cells located in the sensory epithelium of the inner ear. And there are otoliths that are activated. These hair cells are deflected, and you have activation of your vestibular nerve. And this is how balance is achieved. So for example, if your head is upright, just feeling the effects of gravity pulls down on these neurons, and there's a steady stream of action potentials. As you force, or as you tilt your head forward, you force these otoliths forward. The hair cells bend toward the canicium, and you get depolarization and increased action potentials. As you tilt your head backwards, away from the canicilium, there's hyperpolarization, so there's shutdown of your action potentials, leading to decreased frequency of firing. Now the crista ampullaris, we talked a little bit about that, but we can see this is present in the macules. The crista ampullaris is the sense organ of rotation. So they're found in the ampullae of the semicircular canals of the inner ear, meaning that there are 3 pairs in total, and they sense angular acceleration and deceleration. And it's achieved with movement of this fluid in here called the endolymph. Movement of the fluid then is going to move the hair cells, or the hair cell bundle, the kinesium. I should say the kinocilium. And here you can see the vestibular nerve coming in, innervating this hair bundle. So a dancer, when he or she goes through their routine, really relies on the crista ampullaris of the inner ear and the movement of this endolymph. So the ampullary cupula, which is this activated cell attached to the vestibular nerve, bends with the flow of endolymph, and if you're turning or twisting during rotational acceleration, the endolymph moves in the semicircular canal, but it moves opposite the rotation, and it lags behind due to inertia. But it excites these hair cells. And then when this person stops, the endolymph is still flowing, just in the opposite direction now. And it bends the cupula in the opposite direction from acceleration and inhibits the hair cells. So the neural pathways of balance and orientation. The input is the information about the body's position in space, and it comes from 3 main sources. This is from the vestibular receptors in the vestibular cochlear apparatus, from the visual receptors of the eye, and then from somatic receptors of the skin, muscle, and joints. And these somatic receptors might be proprioceptors or nociceptors, but they signal to both the cerebellum and the brainstem, and then the brainstem throws the neurons into the cortex, into the thalamus, where it is relayed. There are a number of muscles in the eyes, the neck, the limbs, and the trunk that are activated by this output of the vestibular nerve. Okay, cochlear implants. I just want to say a couple of words about this. But these are surgically implanted neuroprosthesis, where you have 2 different components. There's the outside component, is generally worn behind the ear, but it's a sound processor containing microphones, electronics, digital signal processing, the battery. And then there's an inside component, which is the actual implant as a coil to receive signals, electronics, and an array of electrodes which are placed in the cochlea. And these simulate the cochlear nerve. And this is how this little boy is able to hear. You can see the outside component. You can't see the inside component, which has been surgically implanted. And it allows for him to have sound perception. Okay, that is the end of the chapter. I'll see you in class.